So now I'd like to introduce our last speaker, Professor Jim McCluskey. Professor McCluskey is a former Professor of Microbiology and Immunology and currently Deputy Vice-Chancellor Research at the University of Melbourne. He has an international reputation for his research in basic and clinical immunology and is recognised for his leadership in the field of HLA and the biology of the major histocompatibility complex. In recognition of his many achievements, he was recently elected a Fellow of the Australian Academy of Sciences. Tonight, genetic control of immunity, trailblazing personalised medicine.
Thank you, Robin, and thank you all for being here so late in the evening. I'm going to talk about how the genes control immunity. Part of what I will talk about has been mentioned already. And I'm particularly focusing on a cluster of genes known as the major histocompatibility complex. It's found on chromosome 6. It's only 4 megabases long, which is about the size of the DNA in E. coli. And these genes control immunity. And I'd like to talk about the role of these genes in the context of personalised medicine. There's a lot of talk around medical circles at the moment about changing the paradigm of medical treatment from being a sort of one-size-fits-all, you've got this disease, these are the pills, take them, to a very much more customised approach. And this is called personalised medicine, where you first of all investigate the genetic background of the person, and then you customise the treatment to suit their genetic background. And that'll be different from other patients, so you end up with every patient effectively being immune. And this is already happening in cancer, where the particular cancer genes are first tested and the treatment is tailored to that particular defect. In some forms of epilepsy, in some forms of diabetes, you have fewer side effects, the treatment is often better than the one-size-fits-all option, and the hope is it's eventually cheaper. And doctors have discovered personalised medicine, and editors of journals write about it with a sort of breathless insight. And in fact, it's actually been around for a long time. And there's no greater expression of personalised medicine than transplantation, where the patient and the donor need to be genetically matched if the transplant is going to take. And this old picture, taken from hundreds of years ago, shows two saints, Cosmas and Damien, miraculously transplanting the black leg of an Ethiopian onto a white patient. Of course, we know this couldn't take. We know that the genetic differences between the black person and the white person would lead to rejection of the organ. And we know that that can occur very rapidly and experimentally. Glenn showed you a minute ago just exactly how Peter Mennewer and others showed years ago that you transplant skin from one genetically distinct person onto another, and then within 10 to 14 days it's rejected. And if you repeat the transplant, it's rejected even more quickly in the memory recall response characteristic of transplantation rejection. And we know that the rejection itself is mediated by these small white cells known as lymphocytes, which rapidly infiltrate the graft bed and start killing the tissue and killing the vascular elements inside that tissue, so it actually goes black as its blood supply is choked off and there's hemorrhage and clotting inside the graft. So graft rejection occurs because of genetic differences between the host and the recipient. Just like blood transfusion reactions occur when blood group matching is not done properly and the antibodies of the recipient react against the red cells that are being transfused from the donor. But the molecules that control transplant rejection are not blood groups. They're a separate set of molecules. They're found on all tissues, not just red blood cells. And they're called histocompatibility molecules, histone meaning tissue, compatibility meaning what it sounds like. And they were discovered on white cells, not red cells. And so they're called human leukocyte antigens, leukocyte meaning white cell. And they're found on all tissues, cells that contain a nucleus. And you get two sets of these from your parents. one set from dad and one set from mum over here, and both sets are expressed on all of our tissues. So we are a classic cross between both parents expressed in the form of our histocompatibility molecules on all tissues. And that's why, as Glenn said earlier, if you tried to transplant this individual with the if you tried to take an organ from this individual and transplant it into the parent, one half would be a match, but the other half would not be a match, and the organ would be rejected. So, how exactly did these molecules work? Well, the fascinating thing about these molecules is they were discovered originally in transplantation biology as the gene cluster that controlled whether an organ was accepted or not. But there were a whole other bunch of scientists working independently on the genes that controlled whether you made an immune response to an antigen or not, to a foreign molecule or not. So these people were working in guinea pigs, and they were immunizing the guinea pigs with small molecules, and only some guinea pigs made antibodies and others didn't. And they worked out that the control of whether you made the antibodies or not lay with these molecules, the histocompatibility molecules. So you had two different parts of science coming together. One group that considered these molecules control transplant projection, and another group that said, no, they don't do that. They control immunity to foreign agents. And of course, they are in fact the same thing, that they were doing the same thing. They were driving the rejection of the transplants because the transplants were foreign, and just as they control whether you make an immune response to a certain virus or foreign agent, they were treating that as being foreign in terms of the immune response. So in about the 1980s, we realized that the immune response genes, as one set of scientists called them, and the histocompatibility genes, as another set of scientists called them, were the same thing. We worked out that if you were going to do transplants, it was important to make sure that the donor and recipient had precisely the same HLA molecules and genes if you wanted the graft to survive a long time. And this actually shows the survival of grafts that are only different by one mismatch versus grafts that are different by 6 mismatches. And this is cumulative graft survival on the vertical axis. So you can see you'd much rather have just one mismatch than to have 6 mismatches if you want your kidney to survive and to remain off dialysis. So this is personalised medicine. This is customising the therapy, the graft, to suit the genetics of the patient, the HLA. And we've been doing it for 40 years, but the editors of journals have only just discovered it. So how does this work? How do these histocompatibility molecules drive graft rejection on one hand, and on the other hand, control what we react to when we mount immune responses? And they do it by showing off small fragments of antigen to the T cells of our immune system, to the conductors of the immune orchestra. So here's a T cell, and on its surface it has a receptor, a specific receptor that will only fit a certain combination of your HLA molecule and a fragment of foreign protein called a peptide antigen. So this is a very strange way of recognizing ourselves or foreign agents, to look at just a small part of that agent. Now the crucial element of this is that we all do this differently, because our HLA genes differ from each other, so that no two people in the room have the same HLA genes, unless there are identical twins in the room. And that is called polymorphism. Polymorphism means variation in gene expression due to mutations in the DNA. So here are a bunch of different apples. Apples are one species of fruit. They're polymorphic. They can be sweet, they can be sour, they can be pink, they can be green. That's all controlled by genes. Well, HLA genes differ in the same way, and so that they have multiple forms of the same gene, and we call those alleles. And polymorphism is simply the existence of different alleles. Now, why is that important? important because the variation from one individual to another in their HLA genes all occurs in this little cleft region of the molecule where the piece of foreign peptide antigen would normally sit. And so the polymorphism changes the chemistry of the cleft so that each of us has a slightly different shaped cleft, so that if you looked into the cleft in a space-filling model of the molecule, you see all these pockets, these nooks and crannies, which accommodate the structure of these peptides derived from foreign agents. And if the shape of your cleft is different from the shape of my HLA cleft, then the type of peptides you will bind will be different to the ones that I bind. It's a very demanding concept, actually. So just pause for a minute. Imagine that on the surface of a cell, we have hundreds of thousands of HLA molecules, each of them bound to a short peptide. The peptide has come from inside our own body. If we're not infected, it's a self-peptide. And so we are continuously showing off to the immune system a vast array of different yellow peptides sitting in the cleft of our HLA molecules. We have what I call an immunological barcode of self that we're showing off to our immune system continuously in health and disease. The barcode that I show off is unique to me because my HLA molecules differ from yours. And so the barcode that you show off to your T cells is different to the one the person sitting next to you shows off. So imagine, for instance, that we have a big jigsaw puzzle. And that jigsaw puzzle is our total set of proteins. And I might say, I'll take that piece, and that piece, and show it off to my immune system, because those pieces fit my HLA molecule. But you would say, well, I'm not going to bother with those. I'll take that one there, this one here, and that one there, and show that off to my immune system. So we each have this different immunological signature in the form of these HLA peptide complexes that we show off to our immune system. And our T cells don't react to that signature. They're educated not to attack our own tissue. They're educated to say, that signature is self, I won't touch that. The only time they react is if instead of there being a self peptide in here, there's a foreign peptide from a virus or a bacterium or a parasite or from a transplant or from a transplant molecule. And so the T cell, on seeing a foreign peptide, receives a signal through its receptor, gets activated, makes nasty compounds which lead to the killing of the target cell or some other function which provides you with immunity.